Alternaria habit and habitat. Alternaria is represented by about 50 species. Several form species are found as saprobes on dead and decaying plant parts and in the soil, while some form species are facultative parasites, infecting a large number of higher plants. The conidia are picked by the wind from the soil and invade laboratories and houses. In laboratories, conidia contaminate the cultures, and in houses these conidia are responsible for allergies, skin diseases and hay fever. The most commonly occurring disease of potato early blight is caused by Alternaria solani. Symptoms of Alternaria. Alternaria shows the symptoms of blight. Early symptoms appear in the form of yellowish-brown spots on the leaves, which enlarge in size and become round to form the concentric rings. If we study these spots with the hand lens, they appear like the target boards, and hence the symptoms are called target board effect. Fig. 1. In severe infection, entire lamina, petiole, stem and even tubers are badly damaged. Edible parts of the tuber turn brown. Mycelium of Alternaria. It is not large and extensive but is short, septate, branched, light brown but becoming darker with age. The colonies of Alternaria are woolly but more compact, with the underside very dark colored. In the parasitic species, the hyphae are intercellular at first, but later penetrate cells of the invaded tissues and thus become intracellular. The cells are usually multinucleate. Genus of Alternaria. The form genus Alternaria occurs universally. It has several form species. Many of these are encountered with as contaminants in laboratory media and on Petri dish cultures. In nature, the Alternarias grow mostly as saprophytes on plant debris and dying plant parts, and also in the soil. Several form species are parasitic on plants. The important plant pathogens are 1. Alternaria solani. It causes early blight, not to be confused with late blight caused by Phytophthora infestans, of potato and other members of family Solanaceae. 2. Alternaria brassicae and A. brassicicola cause leaf spot disease in crucifers such as mustard, cabbage, cauliflower, and knoll cole. 3. Alternaria tenuis. It is the causative agent of leaf blight of wheat seedlings. Agarwal, 1959, reported that A. zinniae parasitizes Ageratum conizoides. Vegetative structure. The mycelium is endophytic, profusely branched and septate. In parasitic species it is both inter- and intracellular, geniculate, light brown and without haustoria. Each cell of the hypha is usually multinucleate. Reproduction in Alternaria. Alternaria reproduces only by conidia, which are produced at the tips of conidiophores. The endophytic mycelium grows out as erect and aerial hyphae through the stomata or ruptured epidermis of the infected host tissue. The conidiophores are short, dark-colored, aerial, septate structures and cannot be easily distinguished from the somatic hyphae. A conidium develops as an apical bud from the uppermost cell of the conidiophore. The young conidium first divides by transverse septa and some of its cells divide by longitudinal septa. Conidia with transverse and longitudinal septa are called muriform or dictyospores. Conidiophore gives rise to one or two conidia exogenously at the tip, but in synthetic media chains of conidia may be produced. Fig. 3. Sometimes branching of the conidial chain is also observed. Any lower seal of the condium produces a bud which develops into conidium. A mature conidium is multicellular, obclavate, elliptical or beaked, about 30 to 12 micro long, 12 to 30 micro broad, and has transverse and longitudinal septa. It is surrounded by two-layered wall, of which the outer wall is pigmented and the inner wall is hyaline. The conidia are readily disseminated by wind. In the presence of moisture and suitable temperature, each conidium germinates by producing 5 to 10 germ tubes at a time. The germ tubes infect the host plant through stomata or epidermal cells or injuries caused by insects. The perfect stage of Alternaria belongs to Plespora infectoria of loculoscomycetes fungus.